This is a pre-lab lecture video for experiment 5. So what we're going to do in this experiment is we're going to do a steam distillation followed by an extraction to isolate the two major volatile components that make up cloves, or what we'll call in a minute the clove essential oil. So the two major components that we're going to isolate are called eugenol and eugenol acetate, and their chemical structures are here. And what you can see is that they are, for the most part, fairly non-polar molecules. Neither of these will dissolve in water. And so we're going to take cloves and we're going to steam distill out what is the essential oil of the cloves. Now, to put this experiment in context, this is actually done all the time to prepare essential oils of plants, flowers, fruits, roots, and those essential oils are essentially what makes up your perfumes and your colognes. And so this slide just kind of gives you the idea of essential oil. The essential is from Latin, which means that it's a liquid that is basically volatile, and then if you say volatile oils, that's Latin to fly. So there are lots of essential oils that you can get from different plant materials. And one of the simplest methods is simply to do what's called cold pressing, where you take the plant material. You can do this with orange peels. If you take an orange peel and you basically squeeze the oil out of it, you can get orange peel oil. And that's called cold pressing. The disadvantage of this is that you get every oil in that plant out when you squeeze it. And steam distillation is a little more selective than that. So the way that essential oils are typically isolated is by steam distillation, which is what we're going to do today. So we're going to take and we're going to place cloves and water in a round bottom flask. We're going to set up the apparatus, this time with a Claisen head and a separatory funnel, and then the rest of it as we set up in experiment 2 with our distillation head, our thermometer, condenser, vacuum adapter, and then we're going to collect things in a 100 milliliter graduated cylinder. We're also going to use the Bunsen burner this time to heat the round bottom flask. Water has a huge heat capacity, and so we have to use a pretty significant heating source. So we're going to go ahead and we're going to use wire gauze, an O-ring with wire gauze on top of it, because we never heat a round bottom directly with a Bunsen burner. We always have to protect that with a wire gauze. And there'll be a video to show you how to set up, or to show you a picture of this apparatus, so that you can see exactly how we will be setting it up. So there's lots of different individual organic molecules that are in essential oils. Some of these are called terpenes, because as we'll learn later on, you can do Diels-Alder reactions to form these terpenoids. So you can see that these are these are common in a lot of essential oils. They are all non-polar in this case because they're all hydrocarbons. Here are some other ones that have aldehyde and alcohol functional groups. Geranol, citronella. The citronella you may come in contact with because that's used in the candles to keep mosquitoes away. You have limonene, which is the major component that is in any kind of citrus oil. Menthol, you find those in cigarettes and cough drops. Carvone is in dill seed and spearmint. And then camphor is a compound that's in like pine needles. And so all of these are just the natural materials that can be used to make up an essential oil. And again, we're going to collect the essential oil of the cloves. Now, there is a newer process that is being used to isolate materials, essential oils, and I'll just mention this in passing. One of the problems that you have when you do steam distillation is that if you have oils that are that will decompose in heat, doing steam distillation and heating the solution up to 100 degrees is not the best method to isolate those. So another method that we use takes account or uses what's called carbon dioxide and supercritical carbon dioxide. In general chemistry, you learn that basically the critical point of a material was a point where the liquid and gas phases became indistinguishable, and that's at fairly high temperature and pressure. And so people will actually extract the essential oils from plant materials by simply passing through CO2 at a higher temperature and pressure so that it becomes this supercritical fluid. And if you pass those then through the plant materials, eventually you can get to the point where you can release the CO2 and you'll just have the oils left over. And this works very well because CO2, if we write out its Lewis dot structure, CO2 is a non-polar material, and so it's able to actually dissolve non-polar materials like limonene and menthol and camphor. And the temperatures that are required for the supercritical CO2 are much lower, and so you don't run the risk of decomposing your materials. And so if you purchase essential oils, you sometimes see that some of them have been extracted by the supercritical CO2. And there's lots of essential oils of different materials. We're going to isolate today the essential oil of clove, but you can do this with cinnamons. You can do it with a number of other plants like lavender, mandarin, which would be the orange peels. And so these are sort of the essential oils that make up the building blocks that you can use to make perfumes and other fragrances. And some of these are fairly common, things like jasmine and lavender and peppermint. Some of them are a little bit more unique and more specialized. So the last sort of background for this is the idea that when you make a perfume or a cologne, you mix essential oils and sometimes individual components, whether they're naturally occurring or synthetic. But essential oils are mixtures of different individual components. And for instance, jasmine oil contains 900 different compounds, and so far only 300 of them have really been isolated. And if you're going to make a fragrance out of it, there's roughly 150 of the raw materials that you can use to create a fragrance. And typically a perfume or a cologne, depending on its complexity and price, will have anywhere from 10 to 15 different components that you can measure. And we're going to analyze our essential oil by GC. But next semester, you'll learn about the technique called GC mass spectrometry. And actually, I've had students do research projects where they've looked at different perfumes and colognes and their different components to mostly look at authentics versus knockoffs to see if we can tell or come up with a method to authenticate those types of samples. But what we're going to do today is we're going to take clove buds and we're going to go ahead and we're going to isolate the essential oil of those, which is going to show two primary components, eugenol and eugenol acetate. We're just going to use the clove buds as is. We might get more material if we were to grind these up in a coffee grinder, but the difficulty is that the powdered material has a tendency to foam when it distills, and so it's kind of difficult to keep the mixture from boiling over. So we're just going to go ahead and use the cloves, the clove buds. So what we're going to do is we're going to do what's called internal steam distillation. I'll show you that in a moment. So what happens in internal steam distillation is that the volatile oils of the clove are nonpolar and therefore they are insoluble, or what we call immiscible, in water. When the solution boils then, you know that the boiling point is when the vapor pressure of that solution equals atmospheric pressure. Well, the pressures of the solution, the partial vapor pressures, are additive, so that a majority of the vapor pressure is due to water, but the small portion of that is going to be due to the vapor pressure of the volatile oils. And so actually what will happen is that as the water boils, it will carry over those vapors, the vapor phase of the volatile oils, and then will condense both of those. In a steam distillation or in any kind of distillation of two immiscible materials, the boiling point of the mixture is always lower than the highest boiling point of the component. You're going to want to make sure that you measure the boiling point of the mixture that you do today or that you do this week, and that boiling point should be less than the 100 degrees Celsius boiling point of water. So the water vapor and the volatile oil will distill together. When the solution condenses, since the volatile oils and the water are immiscible, what you'll see is a cloudy, milky distillate, because what you have are two insoluble liquids that are condensing together. So you're going to set up this apparatus. Again, there'll be a picture on the website showing exactly how you're going to set this up. We're going to use a Bunsen burner to heat. What you want to do is set the apparatus up by setting the Bunsen burner on the bench top, then putting an O-ring in place, then the wire gauze, and then leave yourself about a 1 to 2 inch gap here between the round bottom and the wire gauze, which again has the O-ring attached to or holding it in place. You're going to add a couple of boiling stones, then you're going to add cloves and water, and then the Claisen adapter, the separatory funnel, the 125 should have a ground glass joint to attach it with. All of the ground glass joints are going to be wrapped in Teflon tape. You've got the distillation head, the thermometer with the neoprene adapter, then the condenser, the vacuum adapter, and then you're going to have your 100 milliliter graduated cylinder. Make sure it's clean and dry. You're going to hook up your water line so that water goes in the bottom and out the top as it did last week. And you're going to add 1 bucket of ice, 1 bucket of water to the recirculating systems. Okay, so you're going to set this apparatus up, and then you're going to go ahead and you're going to begin to do the distillation. You want to distill the liquid as quickly as possible. What's going to happen is the vapors will travel up the condenser or up the Claisen head, and we have this Claisen head in here to be able to add more water as well as to keep the material from foaming over. So you're going to have your vapors then turn into the condenser, and what will drip out will be a mixture of the oil and water, so it'll be very milky in color. And so what we're going to do is we have to kind of control the amount of water that's in the round bottom. Too little and we'll burn the cloves and not steam distill them. Too much and the solution will foam over and we'll have to start all over again. So the idea here is that for every 10 milliliters of distillate that we collect, we're going to add 10 milliliters up here and then add that to the round bottom from the separatory funnel. So that way we keep a constant amount of water in here so that it doesn't get too dry and doesn't foam over. So every time you collect 10 milliliters of distillate, you're going to add 10 milliliters to the round bottom to replace that. You're going to collect a total of 50 to 60 milliliters of distillate. The more distillate you collect, the more product that you'll get, but a minimum of 50 milliliters. So that's going to be how we do what's called the internal steam distillation. Once we collect the distillate, then we're going to have to remove the essential oil from the water. At the end of the experiment, you're going to go ahead and break this apparatus down, rinse it out with acetone. There'll be a funnel that you can go ahead and pour the cloves into, because we don't want to pour the cloves down the drain because they'll clog up the drain system down in the basement. Okay, so you're going to distill, collect every 10 milliliters you collect, you add 10 milliliters of water, and you collect to a total of 50 to 60 milliliters of solution. Now, when you get that distillate, then it's it's going to be milky at first, and then it will separate so that you'll see a very thin layer of clove oil on top of the water. And so we have to extract that clove oil out of the water. And so we're going to do that by using a separatory funnel and using dichloromethane. So what we do is we take a separatory funnel, and there's going to be a video posted online to show you how the basics of extraction. But what you do is you pour your distillate into the separatory funnel, making sure that this stopcock is closed. And normally we use an O-ring. We kind of hang the separatory funnel in the O-ring. Okay, they don't put it in a beaker or anything like that. Put it on the O-ring, and we'll show you that in the video. But we're going to end up with a very thin layer of the essential oil on top of the water. And so there's no way to remove the water and leave the oil behind, so we have to do extraction. What we're going to do is we're going to add dichloromethane, which is a polar solvent, but it's not polar enough to be soluble in water. It's a halogenated solvent, so its density is greater than 1 gram per mil. And so when we add dichloromethane to our separatory funnel, what we're going to get is we're going to get the dichloromethane layer on the bottom and the water layer on the top. And the oil, the essential oil, is going to dissolve into the dichloromethane, so that in the dichloromethane layer we're going to have the oil extracted from the water into that. Now, in the video I'll show you how we do this extraction. Typically we do 3 extractions whenever we're trying to remove something from water, because the first extraction will remove so many %, and then each additional one will get the remaining materials. So when we have this dichloromethane solution, when it's heavier than water, which it will be in this case, what we simply do is we drain this solution out into our Erlenmeyer flask. Okay, we pour that into this Erlenmeyer flask. Then we'll add a second portion of dichloromethane. And again, we'll mix this solution up, which we'll show you in the video. And then we drain that second portion of dichloromethane into the same container. And then we do a third extraction. So at the end, we'll have our dichloromethane oil solution in one Erlenmeyer flask, which we then have to dry because it's come in contact with water. And so we dry the sample by adding, in this case, we're going to add sodium sulfate, anhydrous sodium sulfate pellets, well, not pellets, powder, to the mixture, and that's going to basically form the sodium sulfate hydrate, which will then remove the water from the liquid. And so we'll end up with dry, water-free dichloromethane and essential oil. Okay, and I'll go over this in the video as I show you how we do the extraction, followed by how we dry the solution, and how we know when we've added enough sodium sulfate to dry the mixture. That will all be in that video, so please watch it. We're then going to remove the dichloromethane from the solution and leave the oil behind. The oil is not that volatile. Its boiling point is well over 150 degrees Celsius. The dichloromethane boiling point is about 40-ish degrees. And so what we're going to do is we're going to use a rotary evaporator to basically remove the dichloromethane from the solution, to evaporate it. Sometimes people will blow air on the dichloromethane to get it to evaporate. Other times you could just put this in a hot water bath and allow it to evaporate that way. But to do this quickly, we use what's called the rotary evaporator. And so what you'll do is you'll transfer your dichloromethane solution into a round bottom flask that we'll then put on the end of the rotary evaporator. The rotary evaporator has several parts. There's the rotary evaporator itself. There is a pump that has ice water in it, and that basically cools the condenser, which is up here. And so, and we also have then over here, A is a vacuum pump. And so what happens is that we pull a vacuum, or we apply a vacuum to the round bottom that contains our solution. Since the boiling point of a liquid is the temperature where you achieve 1 atmosphere of vapor pressure, that's the normal boiling point. If you reduce the pressure on the liquid, you then reduce the temperature needed to make that liquid boil. So that if we have dichloromethane in our round bottom, by applying a vacuum, we can make this boil very easily at room temperature. And if you don't like that explanation, basically the vacuum pump sucks the solvent off of the solution. What will happen is that this is called a rotary evaporator because this turns around. The thin layer of film of liquid that forms in the round bottom is actually where the solvent is pulled off of. And so what we eventually do is we eventually rotary evaporate. We remove the solvent from the solution until we're left with a very thin layer of oil in our round bottom flask. Okay, and so the TA will do this for you. Since this system is under vacuum, you want to make sure you always have your goggles on whenever you're near the rotary evaporator. This green mesh will, in theory, catch all the parts if it was to implode under the vacuum, but we always make sure that we have our goggles on just in case something happens. So we're going to rotary evaporate the solution. In our round bottom then, we will see very little liquid because steam distillation only produces a very small quantity of essential oil. Once once all the dichloromethane is evaporated, we then take that oil and we're going to add some more 1 milliliter of dichloromethane, and we're then going to transfer that solution to a small conical vial. The conical vials sort of look like this. They're very small and they will contain 1 milliliter of the liquid. So we'll add a milliliter to the round bottom, swirl it around, and then take a glass pipette and transfer it to this conical vial. We may take a second milliliter and rinse and transfer, so that way we've transferred the oil to a smaller container. And then we'll put this conical vial on the rotary evaporator, and we'll again apply a vacuum with maybe a little bit of heat to then rotary evaporate the dichloromethane off, and that'll leave us a very, very small amount of oil in our conical vial. And so that oil then is what we're going to determine the mass of, and then we're also going to do gas chromatography. So before you transfer your solution from the round bottom flask, you want to determine the mass of the conical vial, and then that way, once we're done rotary evaporating, then we just put the conical vial back on the balance and we'll get the mass of the oil that we formed. And it'll be barely enough to see, and it might be kind of a light yellow in color. If you were to smell the vapors coming off of this, it will smell like concentrated clove oil. One of the things you have to be careful of when you make the clove oil is that the clove oil is actually a pretty potent anesthetic, meaning that in the old days they used to take a clove if you had a toothache and you would kind of like suck on the clove because it would numb the toothache. If you get a drop of clove oil on your hand and you touch your face or your lip, then basically you'll have that numbing sensation of that. So you want to make sure that once we get the concentrated clove oil that you're careful in terms of, well, you only have a couple drops, so you don't want to lose those onto gloves or anything else. Okay. Now, why did we dry our sample before rotary evaporating? Well, if we had not dried our sample by adding the sodium sulfate, which sodium sulfate reacts with water. If you take anhydrous sodium sulfate, which is a solid, it will actually react with water to form what you learned in general chemistry, which are the sodium sulfate hydrate, which is also a solid. And so adding sodium anhydrous sodium sulfate will then react with any water present to form the hydrated sodium sulfate. And so then we can simply decant that solution away from the sodium sulfate crystals, or if the worst case scenario is we just have to filter the solution. But normally we can decant the liquid from the solid. But why did we dry this solution before we put it on the rotary evaporator? Because the rotary evaporator isn't going to be operating at heat to evaporate the water. And so if we don't completely dry our solution, when we get our oil, we'll see the oil and then we'll see these other droplets of water in it. That's going to make our mass too high, and it's going to make our oil then very wet. So we want to make sure that we do a good job of drying the solution before we rotary evaporate. And every time we use a rotary evaporator this semester, the first step will always be to dry the solution, decant the liquid from the drying reagent, and then distill or rotary evaporate the solvent off. If we don't, we'll see water droplets in our product, and we don't want to do that. So that's why we dry. Then we're going to take our oil and we're going to use a gas chromatograph to determine the percentages of eugenol and eugenol acetate in the oil. These are the old GCs. They've been around for probably 50 years, but they still work very well. And we're going to use these GCs to analyze our product. And so just like you sort of did in general or chemical principles lab, we're going to inject 1 microliter of our oil into the instrument. This is our injection port. The sample will then be vaporized. The vapors will then travel through this column that contains a stationary phase, and the components will separate on this column according to boiling point. Eugenol has a lower boiling point than eugenol acetate, and so eugenol will go through the column first, followed by eugenol acetate. Once the vapors come out of the column, they're detected, so that what we end up with are peaks that show us, for instance, the eugenol will come out first, and then a smaller peak for the eugenol acetate. If you see a peak very early on, this is dichloromethane solution or solvent. And so if we don't completely rotary evaporate our sample to dryness or to find that oil, we'll see a little peak here due to the dichloromethane. So the peak then comes out at a specific retention time. Retention times vary depending on the column that you use, the temperature of the oven, the temperature of the injection port where you're injecting your sample. So retention times can actually vary. And so what we do is we have authentic eugenol and eugenol acetate, and so we'll inject those standards into the instrument to determine what retention times we see for pure eugenol and pure eugenol acetate. So this is a typical chromatogram that we'll see. If we don't see any solvent, we'll see eugenol, eugenol acetate. The instrument, we're going to collect this data on a computer, and so then what you'll see is you'll see the retention times, and then you'll also get your percentages, and roughly you'll find that eugenol is about 90% and the eugenol acetate typically is 10% of the essential oil. These chromatograms that we're using, these GC instruments, aren't very sensitive, and so I may take the eugenol, the essential oil that we get, and inject it into a much more sensitive instrument. And if I do that, you'll see that there's probably 20 or 30 additional compounds in this mixture beyond just the eugenol and the eugenol acetate. Okay. So that's what we're going to do for this experiment. We're going to set up and do our steam distillation. We're going to do the first extraction of the semester to extract the oil away from the water. We're then going to dry the dichloromethane, rotary evaporate the solvent to leave us the oil, and then we'll analyze the oil by GC. Okay. Now there are going to be 2 and possibly 3 demonstration videos that are below this link. What you want to do is to watch those. One will sort of go over how to set up the apparatus, set up the steam distillation, and there'll be a picture of that. Then how do we perform an extraction using the separatory funnels? How do we dry the sample, including how do we know how much sodium sulfate to add? In a procedure, I'll usually say add enough so that it doesn't clump anymore. We'll show some videos of what exactly that means. And so you want to make sure that you watch those because you will be doing those manipulations during the lab experiment.